Speaker's Corner aboard the Villa Vie Odyssey, hosted by Kit Cassingham.
Good evening, folks.
Hello!
We've got a Speaker's Corner, which is, I guess, why you're here, huh? Will you, I'm going to try something different and ask people to turn their phones and noise-making devices off so that we don't get them shared with, competing with the speaker. And tonight we have, I thought something's, and I'm going to get this wrong. We have Biochemist, this is Dr. Frank Miskevich. He's married to Dr. Catherine, who knows Dr. John. Is Dr. John in the room? No. But he is a biochemist and molecular scientist.
Biologist, but almost.
He's a scientist. He knows his stuff, so listen to him. How's that? But John's given us a lot of very helpful and interesting talks. And I'm looking forward to this one because I think we're going towards Malaria Country, huh?
We are actually in it.
In it. Oh, great. So, Kevin, you said that something about mosquitoes and malaria, so listen up. So, help me welcome Frank.
All right. So, just to give you sort of an overview of what I'm going to be talking about, I'm going to start talking about how common malaria is and what causes it. It's caused by basically a very specific organism that has a very unusual life cycle. And after that, we'll talk about how you can actually not get malaria, which I strongly recommend for all of you as we're going into some malaria-prone areas. For those of you who already had malaria, congratulations, you survived and tried to avoid that. 2 people have come up to me already and said on the ship just recently that they've had malaria. So this is a disease that affects a lot of people. And then finally, I'm going to talk about how people actually can cure malaria after they've gotten it. And so this is, some of it's very recent, but it has gotten to be a big deal, and it's actually made an impact on how much malaria is present in the world. Right? So basically the first slide is showing the areas most at risk for malaria. And as you can see, it's right through the middle of the equator, basically. So Africa is a huge area, but it's also prevalent in Asia and Southeast Asia. And if we look at this one little tiny island over here called Palawan, anybody know it? Right? There's actually regions of Palawan where malaria is endemic and you're at high risk of getting it. I don't think we've spent much time there, but we're getting into the area of the globe where malaria is a really big deal. If you're looking down into this Papua New Guinea area, Solomon Islands, even down near Vanuatu, Sumba, this is where we're headed, right? And so we want to be careful not to get malaria because it really isn't a very pleasant disease. Globally, you have about 263 million cases of malaria annually, all right? This is a huge disease in terms of number, and it's basically got symptoms very much like the flu, right? But most of these people who are getting malaria are in Africa, like 94% of them, right? 600,000 people die from malaria, 95% of them in Africa, right? But worst of all, malaria is, it's sort of a debilitating killer. You get this sickness that just won't go away, and you can't get rid of it, and it makes you weaker and more susceptible to other things. So the weakest people, usually the children, are the ones who suffer the most. So about, 76% of the deaths caused by malaria are children under 5 years old. So our adults are more able to withstand the parasite, but kids don't have the resources and let's call it the stamina, the endurance to actually deal with this. Okay? So this is a graph showing how many people were dying of malaria over time. And if you look back here in 1980, it was about 600,000 people, with a significant percentage in this red area, which was in Asia. So Asia used to have a significantly higher percentage of malaria than it does now. Africa's always been a big deal, right? A lot of the people who die come from Africa. So as you can see, as the population was growing, the number of deaths went up. And this is where the Gates Foundation came in. So the Gates Foundation, for those of you who don't know, Bill Gates, Microsoft, gobs of money, he and his wife set up a foundation which focused on just a few areas, one of them being malaria. And so in 2000, he set up this foundation and really jump-started how to avoid malaria, how to treat malaria, how to cure malaria. And so if you look at it, this is when the deaths were up in this area, sort of leveled off right after that, and after that it's been decreasing most of the rest of the time. Ever since about 2020 or so. Gee, I wonder what happened in 2020 where people didn't care about malaria anymore, right? Yeah, COVID hit, people stopped dealing with malaria, you have a spike, right? Then deaths basically returned to roughly where it was, sort of on the same downward trending line. This data was from 2023, and so the deaths peaked a little over about 900,000 in 2005 or so. But one of the big changes was the amount of malaria in Asia has gone down significantly. And I think that's due to a number of things, especially Asia becoming wealthier, a lot more money in I'm going to call it the greater region, but also the development of a particular drug in the early 1970s that I'll talk about in a bit, right? So, what causes malaria? One thing, Plasmodium. It's basically this tiny cellular parasite. It's a single-cell eukaryotic organism that basically has a number of relatives, almost all of which it cause diseases, right? There are 6 species of Plasmodium that cause malaria, and they're all carried by this one mosquito, Anopheles, right? And so basically, you can avoid mosquito bites, you can avoid getting malaria. Wouldn't everybody like to be able to get rid of all mosquitoes in the world? Right, it's not that easy. Okay. But basically, the parasite is a fairly complex organism that lives in the blood, or at least the symptoms of malaria are caused by this parasite when it gets into the blood. And if you stain it, you can actually see it sitting in these red blood cells. And this is one of the ways they'll diagnose it. Again, depending on what kind of pictures you're taking, you can get different images, but you're basically looking for these bumps, these forms of malaria that are inside the red blood cells. They'll reproduce, grow, lyse, infect more red blood cells, infect liver cells, infect mosquito cells. So, symptoms of malaria, if you're not used to treating it, you'd never know it was malaria, right? It has a lot of the same symptoms as the flu. You've got a fever, you get coughs, headache, muscle pains. Anyone here coughing a lot at various times? Having a fever, right? Theoretically, if you were in the right area, that could be malaria. So it's not something you would go to the doctor immediately over unless you're really worried about the disease, right? Because if malaria progresses and gets worse, then you start suffering more severe symptoms. Oh, before I go on, one of the classic symptoms of malaria is what's called paroxysms. Basically, you go from feeling hot to cold to hot to cold to hot to cold, and it varies in basically a 2-day cycle, which is the amount of time it takes for these parasites to grow, lyse, and start infecting more cells. So they infect more cells. I think you get a fever. Once they're inside, it goes away. You feel this chill. Then they lyse the next cell. You go through these cycles. Okay. But when you get severe malaria, this is when people start dying, right? Severe symptoms include coma, convulsions, seizures. Again, these are the things that people say, oh, wait a minute, maybe I should get treated for something here, right? Because remember, a lot of the people who die from malaria are in Africa, and most of Africa is a very poor region of the world, so they're not going to have medical care easily available, okay? So Plasmodium itself is a eukaryote. So even though it's the size of the cell, it's not a bacteria. Antibiotics don't do crap against this disease in general. One exception, I'll talk about that bit. But it's a eukaryotic organism. So basically the world is divided up into two kinds of creatures. Things with a nucleus, things without a nucleus. The prokaryotes are your standard bacteria. And they have a number of other differences, and these are what antibiotics typically target. You know, we don't have any cell walls. If you can interrupt cell wall synthesis with something like penicillin or that entire class of drugs, you can stop the replication of those bacteria. Right? If you can affect the ribosomes, which are different in prokaryotes, you can kill bacteria with that kind of an antibiotic. Right? Problem is, these have basically the same organelles that you and I have, right? They have a nucleus, they have a Golgi apparatus, endoplasmic reticulum, mitochondrion, right? And so these have a lot of the same cellular structures as we do, making it a lot harder to kill parasites, right? Because you have to be very careful. You can kill the parasite really easily. Problem is it kills your cells too. Usually not a good idea. Right? And so trying to target these organisms, you typically have to look for something that's unique about them, something that's different. And we're going to talk about one of the things that they target. Right? But this is an organism that actually has a really complex life cycle for something so small. So it not only requires humans, but it also requires the mosquitoes in order to reproduce. So basically, the mosquito has the parasite growing in it. When it bites you to suck your blood, some of those parasites go into your bloodstream and they start replicating. They infect primarily red blood cells, but they also have a separate form that infects the liver. And so when these organisms are growing in your body, they're dividing, rupturing the cells, infecting more cells, rupturing, blah, blah, blah. Right? This is what causes the disease. What happens is when you're infected with this disease, another mosquito comes and bites you. It picks up some more of the malarial parasite, and then it goes into the mosquito to complete its life cycle and start infecting more people. Right? So it actually requires two hosts, a mammal and the Anopheles mosquito. 5 of the 6 species only infect humans, right? Some ways this is good. Okay? The other species is zoonotic. Zoonotic meaning that it's able to infect other organisms. And so one of the ones that it can affect are macaque monkeys, right? So, say you have a disease in the monkeys that might be able to infect humans. Can anyone think of a disease that originally came from monkeys in like the 1930s? Virus. What is it? Not quite monkeypox, but it would be HIV. Right? This is what came from Africa. It was a very slow developing disease, spread around the world. It takes, 10 years often to affect people. And so there was a long time before this could develop. Mosquitoes could easily carry out that transfer of the virus from monkeys to humans. And this is how diseases get born, really. Same thing with the COVID virus, the SARS, several other different things like that. They come from one organism, They go through some kind of change, then they can affect another organism, right? One that they're worrying right now about is bird flu, right? Coming from birds, a flu virus that infects birds, spread to cattle, spreading to humans, and it's getting worse in a number of areas, particularly in the United States, right? But the idea is you have this mosquito vector that, because it takes blood from different species, is a perfect way of transferring genetic information or organisms from one species to another. Okay? And because of the life cycle of this plasmodium, it's actually hard for the immune system to target. You have a lot of antibodies and things floating around in your blood. Well, that's where you find plasmodium, except it's hiding in the middle of these red blood cells. So we know red blood cells are important, right? Essentially, they're really simplified. I'm going to call them dead cells because they actually don't have much DNA in them. They're basically a membrane sac stuffed full of hemoglobin, and that's almost, and some ribosomes and RNAs in that. But it's designed really just to carry oxygen from your lungs out to your tissue and then pick up carbon dioxide and go back to your lungs, right? And when these organisms are living inside the red blood cells, they're very hard to attack. Antibodies can't get to them. They can attack things that are stuck to the surface, right? There's a set of molecules called complement that helps identify antibodies stuck to the surface of cells. And that's basically one way you can kill it, but when it's stuck inside the red blood cell, it's really hard for things to go in there, and they lack ways of, or the body lacks ways of detecting what's inside them. Right? Liver has the same problem. Your liver is an essential organ. You can't live without one. It's got to be functioning or you die. But it's an immunologically privileged area, meaning that the immune system cells can't really go in there because of the variety of reactions that the liver takes place. It does a lot of purifying of chemicals from the body, and the immune system doesn't do well in that particular area, right? So how can we get rid of this? How can we prevent getting malaria? Insect repellent, one of the best ways of doing this, right? People use DEET, right? How many of you like DEET as an insect repellent? Yeah, this is the one I particularly like. Another one that's apparently very effective is oil of lemon eucalyptus, right? This is a newer one that they came out with. It contains a chemical called PMD, or paramethane diol. Supposedly it's as effective as DEET, right? Except it's not very good for young children. That's one of the specific requests, or one of the specific contraindications, right? But there's also a drug called Picardin, IR 3530 or IR 3535. Wonder where they get some of these names. 2-undecanon, right? So these are particular insect repellents that'll work on people's skin, right? So these are the things that you can apply and then wash off and repeat as necessary. Right? Another really useful one, especially in areas that are prone to a lot of mosquitoes, is this particular chemical called permethrin. It's a very good insect avoidance molecule. Mosquitoes hate it. So people will soak clothes or netting or something in this permethrin, and it basically repels mosquitoes. And you can wash this, you can wash it like 5 or 6 times before the permethrin gets off your clothing. We actually bought some and sprayed our shirts and things with it, and it works. You know, for what it does, it's another good way of doing it. Right? How many of you have seen those mosquito nettings in a lot of countries where there's a lot of mosquitoes? I went to Brazil in Manaus one time, and I woke up in the middle of the night. I was sleeping under one of these mosquito nets, and I have never seen so many bugs on the outside of anything in my life. That was terrifying. I had to go to the bathroom, and I just waited. It was right? Another thing that really helps is if you're wearing long sleeves or loose-fitting clothing. I can tell you a little story involving Catherine. We were hiking in Canada one time, and it was mosquito season, and she was wearing these tight leggings and sort of a tight shirt, and she was getting tired because it was getting a longer hike, and she didn't want to stop because the mosquitoes kept chasing her, and she was pushing forward to keep running away from the mosquitoes. You know, if she wears these tight leggings, if you wear looser fitting clothing, looser fitting shirts and that, it's one way to help keep from the mosquitoes from being able to get through you. If you have thin pants or thin shirt on that's tight fitting, they can go right through into your body, get your blood, and you'll be just as unhappy. Right? And she's remembering this event.
Perfect. I counted the mosquito bites.
But another thing that really helps with malaria is fairly prompt treatment. If you go to a malaria-prone area, travel away from it, they don't know to look for malaria, right? This is where having that recent travel history, especially for us who are living on a ship, going God knows where on the earth, traveling around, coming back to the ship, we're going to be bringing diseases back and forth. It's natural, and I'm not blaming anything. This is just the way diseases are going to spread. And because we're traveling to all these different regions, you know, we are going to get things. And so one of the things you want to do is, if you might have malaria, you do want to get tested before the symptoms become severe. Okay? Now, for people who are going into malaria-prone regions for a short period of time, there are drugs that you can take which will actually prevent the malaria from actually developing. This is called chemoprophylaxis, and the idea is if you can get keep the malaria parasites from getting established in your body, you're not going to get the disease. They'll basically die out, right? Chloroquine was a drug that they used to prescribe frequently as a preventative malaria treatment. Unfortunately, the Plasmodium species developed a resistance to chloroquine sometime in the 1960s or so, right? And so it stopped being useful. Today, there's a drug called Lariam. This is one I've taken in the past as a prophylactic drug to prevent you from getting malaria. Another one is a drug called doxycycline, right? And this is an interesting one because doxycycline is actually an antibiotic. So, a little bit ago I told you antibiotics don't work on Plasmodium. This one does. And this one it does because of a very particular organelle. They don't label it here, but it's this little circle right in here called an apicomplexa, an apicoplast, right? This is actually what defines an entire class of organisms called apicomplexa. They have this apicoplast, and it's a really unusual organelle in that it's surrounded by 4 different membranes, right? So you have the cell membrane. Inside the cell, you typically have, say, a nuclear membrane, and then mitochondria with their own membranes, and chloroplasts with their own membranes. This one has 4 membranes around this apicoplast. And they think this developed specifically because sometime in the very distant past, the precursor to Plasmodium and Toxoplasm gondii, right, these apicomplexi basically ingested red algae, and that red algae worked symbiotically with that earlier organism and literally developed an endosymbiotic relationship. So these apicoplasts have their own DNA, they make their own ribosomes, and it's basically the leftovers of this organism that used to be a free-living organism that provides an essential function for these Plasmodium species. You get rid of the apicoplasts, the organisms die. And this is one of the things that scientists use to try to target this organism. We don't have any apicoplasts in our bodies. If you can find a drug that'll block its function, and that function is essential to this organism, Now you have a target and the drug that you can use to kill off this organism specifically, right? So anyway, that's just a little history of why doxycycline, even though it's an antibiotic, actually can affect this particular organism. Right? And there's another pair of drugs called atovaquone and proguanil. Right? It basically comes as one tablet, and it's a very good chemoprophylactic, right? Because it will block both the liver and the red blood cell forms. Right? Most of the others, the lariam and the doxycycline, are known as suppressive prophylactics. They're only going to work on the blood forms. This is what causes the symptoms, this is what causes the disease, but it doesn't directly get rid of the things in the liver, right? And this is a problem because just like antibiotics, you're prescribed these chemoprophylactics over time. If you're taking them, you want to make sure that you take this dose as long as it's been prescribed for, anywhere sometimes up to a month or so after you've been to these regions, right? Because if you don't, if they have those malaria parasites sort of hiding out in the liver, well, they can grow and then start infecting you, and then you end up having malaria even though you were on this prophylactic substance. Okay? But all of these drugs you can't take if you're living with malaria every day of your life, right? This isn't going to work for the people who are living in the areas, but for visitors to the area, these are drugs that people will actually be able to use and help limit their exposure. And since we have no other forms of resistance to malaria, that's going to be important, right? But, you know, malaria's been around for a long time. There is actually a genetic modification that helps prevent severe malaria, right? These are organisms that focus or that lives a long time in red blood cells. So, most common protein in red blood cells, hemoglobin. You can have a single base pair mutation in hemoglobin that'll reduce the severity of malaria. It reduces malaria severity by about 50% or so. So you'd think this would be a great gene to have, right? Well, it's actually found in about 25 to 30% of people living in these endemic areas, but there's a problem. It provides resistance to malaria if you have one copy of the gene. If you have a second copy of the gene, now you have two of these different altered forms of hemoglobin. Again, it's one specific nucleotide mutation in the gene. One base pair change, it causes a form of hemoglobin which causes the red blood cells to form these sickles. Sickle cell anemia is basically when your parents have the resistance and they come together, you get unlucky, you get two copies of the bad hemoglobin, you may not get malaria, but you have anemia, you have a hard time getting your blood cells through, you get more strokes because these cells, they don't fit through the capillaries very well, right? And so this is a case where malaria is such a big deal to the people living with it that they can lose 5 or 10% of their children to sickle cell anemia, but the resistance to the disease is so important that have this mutation is found very commonly in these people. So malaria is a big deal, right? If you're living in a malaria-prone area, you want to avoid it. Okay? So, how do we know if you get malaria? Gold standard is microscopy. There's a particular stain called Gemsa stain. It basically causes the DNA to turn bright purple. So you have white blood cells with particular shapes, and then you have these little things growing inside of red blood cells. You can see them starting to occur more and more in some areas. Some places there's a big bundle of them. These are the plasmodium that are actually reproducing, and it can affect a significant percentage of your red blood cells. One of the ways that they can tell the severity of the disease is by counting up the percentage of red blood cells infected with the plasmodium. Right? But microscopy takes a certain amount of time. If you're trying to screen hundreds of samples, you have to be pretty good at it. To tell the species from these particular little swirls, you have to be really good at microscopy. And so, even though it's relatively inexpensive, it's not always the best way. It's a very clear way, right? But it's not always easy. How many of you have ever seen those pregnancy tests with those lines coming through? They have exactly the same kind of test for malaria, right? Basically, you have a control line just to show that, yes, the test is working. And then you have, in this case, you have two different additional lines which show different species of malaria. If you've got this single line, you have one species, the most common one. If you have the other one, there is 3 different species it might be. But again, it's a test for what it can be. It works just like a pregnancy test. It's called an enzyme-linked immunosorbent assay, technical name for it. Basically, it's these lines that appear or don't appear whether you have the disease. Right? It's very easy to identify. Right? It's simple. Problem is it costs money. And Africa is not exactly the richest continent in the world, so it's actually not as useful for most people because of that. Right? And again, when you get a positive with these tests, you're also typically going to want to do the microscopy on it to make sure that it's malaria and not something else. Right? Third test that people use, right? PCR. People have all heard of PCR now because of COVID, right? This is one of the things that they use to test for COVID. But what they have to do is they're testing thousands of people. They're sending all of these samples off. They run the machine. They identify the positives, send the information back out, and it takes a couple of days. In a clinical setting, PCR isn't as useful, especially in a place like Africa, right? But what it really gives you is a lot of information about the species, what kind of plasmodium is actually infecting a person. And so basically using a set of what are known as primers that amplify a piece of DNA, you have different size bands that are indicative of the species that you're dealing with. Right? And so this particular one shows 5 different types of species. You have a positive control in there for human. Right? And so this gives you a very clear and unambiguous determination of what species you're dealing with. And in some cases, that species information is important for what drug you're going to use to try to cure it with. Right? So, how do we actually go about curing malaria? Right? One of the earliest treatments in the West was a drug called quinine. Right? The first treatment, they used a bark extract. They basically boiled the bark in water and people would drink it, and it would have treatment, or it would help to protect people from malaria. They developed this in the early 17th century, 1620s or so. Right? They purified the quinine for the first time in 1820, and this was actually the first purified chemical that they used to treat an infectious disease. It wasn't an antibiotic, but it did target this malarial parasite. It's this white powder. Right? But it's not perfect. Right? You have a number of pretty severe side effects from quinine. So right now, even though it still works, people would rather use other drugs because they have less severe side effects. But you have tinnitus, which is that ringing in your ears. It'll cause that. It'll cause vision issues. I think it was Jeff who was saying when he was given quinine for malaria, he had vision issues from it. Right? It can cause deafness, again related to that tinnitus, irregular heartbeat. Right, so it's not exactly an ideal drug either. So people were looking for more drugs. There was a drug called chloroquine. This was developed or discovered in the 1930s, and I think it was pretty heavily used along with quinine during World War II, when all of those World War II battles against the Japanese were being fought and all the islands that we're going to be visiting in the next few weeks, right? This was developed in the 1930s. It was used extensively because it was very effective, but basically in the 1960s, especially in Asia, a resistant form of Plasmodium developed because chloroquine was in such heavy use, and the incidence of malaria started going up again, right? So some of you probably remember the early to mid 60s, right? This is going to be when you have the Vietnam War going on. And the Vietnamese soldiers, just like the American soldiers, suffered from malaria. Well, they had a friend in China, and China started a research program trying to develop a treatment for malaria. And a woman named Tu Yu found an ancient treatment in Chinese medicine that was originally used starting around 340 AD. So basically 1,300 years before the West started using this boiling this bark, right? And it was a herb called sweet wormwood. You can actually buy wormwood extract and oil of wormwood in some health food stores. Okay, there's some side effects and various other chemicals there, so I'm not recommending. This is not a recommendation for wormwood or anything, but it has supposedly some side effect or some health benefits, right? But Tu Yu was a biochemist, and she went through, isolated what was coming out of the wormwood, and purified a chemical called artemisinin. Okay? This is currently the frontline treatment for malaria. This is what they give everybody to actually treat this. Came out in the 1970s, and she was awarded the Nobel Prize for developing one of these drugs that's actually effective against parasites. Interestingly, for those of you who follow sort of drug development, this is the same. There was another group that was awarded the Nobel Prize at the same time as she was. They developed the drug ivermectin, which is actually a very, very good drug for roundworm and flatworm type infections. It's a very good anti-worming drug, right? Not so good for COVID, but it's actually still used intensively for these other diseases. Okay? And interestingly, the way this drug works, I'm not going to go into the structure, believe me, we're not going to talk about detailed chemistry or anything like that, but there's this 2-oxygen bridge in here. The drug gets activated so they can start fighting Plasmodium only when you break this thing apart. And that breaks apart specifically in the presence of hemoglobin. This heme group that's complex to the protein, it provides the right environment to break this bond and make it start reacting with things. You've got lots of hemoglobin in your red blood cells, you've got the Plasmodium in the red blood cells, it kills the Plasmodium. Right? But it has its own issues. Right? It is one of the essential drugs under the World Health Organization, so even out of the way places, you will find artemisinin. It's somewhat expensive because it's hard to prepare, and they don't want to use it by itself. Because chloroquine and there's chloroquine resistant strains of malaria, well, there are certain strains of malaria which seem to be developing resistance to the artemisinin as well. Right? We don't want that. This is their main treatment. We don't want to lose it. And so the World Health Organization strongly recommends that you get a combination therapy of 2 or 3 different drugs that are targeting malaria to try to prevent the development of the diseases. This is the same thing you find now with a lot of antibiotics. They try not to sometimes for certain diseases, They try not to give you just one antibiotic, but they may give you 2 or 3 at the same time so that you can't develop a resistant form. It's not working very well because too many people are overusing antibiotics. But when you start losing your best defenses against particular classes of organisms, whether it be malaria or bacteria or whatever, bad things happen, right? And there's no good way around that. And so these are different forms of artemisinin, again, chemically modified so that they're slightly different, but they all have some of the same problems, right? These drugs are, you can only get so much artemisinin out of this wormwood. From what I was reading, they process about 180 tons of wormwood. This wormwood that they use is basically this little fern-like herb, and so 180 tons of it is quite a bit, right? And then they get the artemisinin. So it's actually very expensive, low yield of the drug coming out of the artemisinin, and so people wanted an alternative form, some other way of getting this drug. And so since the 1980s or so, genetic engineering has been progressing, and there are certain organisms that we, the scientists, like to use. E. coli is a great one, but if you're trying to make something that you find in mammalian cells or eukaryotic cells, you want to use a eukaryote. And the eukaryote of choice is a yeast, Saccharomyces cerevisiae, bread yeast. Basically, it's been widely used for producing various chemicals from biological processes. And what they did is they analyzed how this drug was made, what artemisinin looks like, where it came from, and then reprogrammed the yeast to actually make this chemical, right? So the artemisinin, we're not going through a lot of the chemistry, I'm just giving a really quick summary of what they do, right? But it's a type of lipid called a terpenoid, right? The key thing is, it's made up of these 5 carbon subunits called isoprenes. Again, don't worry about the names. Put 3 of these together, you get what's called a sesquiterpenoid, and these are common in your body. They're attached to certain parts of your cell membrane. They're used to anchor proteins to your cells, and the yeast make these as well. And so this is a very common type of lipid that you find all over the place. Just like steroids. You can have 20 or 30 different kinds of steroids, each one that's going to be slightly different. Well, plants, you've heard of plant alkaloids, those different chemicals found in plants. A lot of them come from these terpenes. And so basically what you do is you have the basic mechanisms for putting these things together in the yeast, right? You can make the basic molecule, but you can't do all the steps because normally what the yeast does, it uses it to make these other things. So scientists go in and say, okay, what enzyme is going to use this drug that we don't, or use this chemical that we don't want to use it? All right, let's go in and mutate it, knock it out so you can't make the side effects, right? If it normally goes on to make this molecule here, block this enzyme, can't make it. Now it's got all this extra, let's call it intermediate, that's sitting around. Okay, what do we need to do? Oh, we need to add this other group to it. Okay, let's put that enzyme in the yeast. Take it from wormwood or something else, put it into the yeast, so now it'll get processed to this next group. Repeat the thing. If it normally goes on to form something wrong, kill that process, give it another enzyme, redirect it so that it synthesizes what you want. And this is where all those if you've had biochemistry or something in the past and they talk about all these pathways and how you make these different things, scientists have gotten to the point where we can go into that pathway and say, we want to make this and not this, so let's block over here and add this enzyme, and you can go through and make these yeast molecules, which grow very easily, right? you can grow up 20 or 30,000 liters of yeast broth at a single go. I've grown 20 liters easily in the lab, right? If you're brewing beer, you grow yeast cultures all the time. We grew 5-gallon batches when we were brewing beer, right? Same yeast, right? You have to go through the purification process, but scientists have gotten pretty good at reprogramming the yeast so you can get more access to these drugs. And there are a number of chemicals that are hard to find in nature, but if you can reprogram something else to take those basic starting units and go along a different pathway, you can do a lot of things with it. Taxol, you may have heard of this. This is a cancer drug that was found in the bark of a Pacific yew tree. They can now make in Saccharomyces by doing exactly the same thing, using the same basic molecules, redirecting it, and using this as a way to develop a hard to refine chemical in something that's easier, where you can get lots and lots of it. So again, this is a relatively recent accomplishment. It's called metabolic engineering, and there's whole research, there are whole review articles about how to do this in yeast, and it's a big area because chemical production isn't always easy. If you have the right enzymes, you can make it easier, right? And finally, because artemisinin isn't perfect, right, it has a number of issues. It's not very bioavailable. It gets broken down very, very quickly by the body. It gets expelled from the body. So you have to get these injections every day. You can't be given a pill for this. It doesn't go through the digestive system, right? It has to get into the red blood cells, so literally they have to inject it into the bloodstream. You don't want to give this to pregnant women, especially early in pregnancy. You can't use it for prevention like you can Lariam and some of the other drugs, because literally the drug breaks down so quickly. This is why they give you, also why they give you a combination drug. One is very quick and very good. The other one lasts longer but isn't as effective, but helps. And so they'll try to give you these drugs at the same time. Unfortunately, some of those additional drugs that they give you don't react well with HIV positive individuals. Their biochemistry is different. The cells start dying. The virus starts replicating. Bad combination, right? And again, resistance is developing to artemisinin. And the more you use a drug and you use it because it's very effective, the more likely you are to develop resistance. Okay? So because kids are more at risk of dying from malaria, scientists wanted a way of being able to help these kids and treat or give them something that would help them ward off malaria. And one of the best ways of doing this is using vaccines. So in 2021, the first vaccine against malaria was approved. This is again funded by the Gates Foundation, who put literally billions of dollars into these projects. But it's specifically aimed for children because the drugs aren't really well tolerated by kids, and kids are the ones that are most likely to die from the disease, right? So over the course of 12 months, if you give them an injection, one of these vaccine injections, you can reduce the cases of severe malaria by about 50%. And if you time it right, so that the vaccine is given just before the rainy season when the mosquitoes are most active, you want the body's reaction to be maximized when the mosquitoes are maximized, you can actually get a 75% resistance to malaria, which is pretty good, right? But a more telling statistic is a 13% reduction, and only 13%, but all-cause mortality in children. So they're not just looking at diseases caused by malaria, right? These are all kids, a 13% reduction in infant mortality up to the age of 5 years old, right? That's the kind of toll that malaria takes on the young. So this one treatment, you can reduce the number of fatalities among children by 13%, which is pretty darn impressive considering the number of ways children can die, right? But because it weakens the kids, they have to fight this thing. They're no longer able to resist other diseases as effectively. And so this vaccine is just starting now to make a difference in their lives. So with that, I'm basically finished. I can answer people's questions up to a point, and I hope you enjoyed it.
Does the ship have the facilities to detect it?
The ship certainly has a microscope. I will bet they have the Giemsa stain that they use for detecting it. So I haven't actually asked, but this is the kind of basic chemicals that they have, and because we are traveling through malaria areas and will be, especially when we start going to Africa, I would hope that they have some of these anti-malarial drugs on board.
Well, and do they have the expertise to review what's under the microscope?
I honestly haven't asked the question. I've done Giemsa staining. It's not that hard. Identifying the details about the parasite, never tried that. But the technique is, for someone who's done microscopy, it's not that bad. And you can find protocols for doing this very easily. I think John has a question over here.
Was gin and tonic effective? And is it still going to be the treatment of choice?
So, John's question about the gin and tonic. Remember the quinine that we were talking about? That's that sort of bitter taste in the gin, or sorry, in the tonic water that gives there. I don't think it is, because quinine is only so efficient at it unless you're drinking an awful lot of gin and tonics, and then you might be having other problems to go with it. But yeah, that's this is one of the places you find quinine, and it is in that tonic water, and that's that bitter taste that goes with it.
Can you get more than one type at a time?
You can. So when you have these Plasmodium species, certain areas are going to have more of a particular species than others. And again, that varies and it changes over years, and I couldn't tell you what species are most common and where. But you can definitely get more than one species at a time. And on those malaria tests I showed back here, right, you actually can have two bands that show up as, if they're both positive, that means you have, you often have a combined mixture of these two different Plasmodium species. Now, fortunately, for the most part, artemisinin and the other drugs are able to target all the different species, at least for the moment, right? But if you develop a resistant strain, then it might work for a while, you stop taking the drugs, you get the disease back, and then that drug is really no longer effective. And that's where resistant strains basically come from. You think you've got it cured, you don't kill everything, but you feel better, so you stop taking the drug, and then you get it worse, and it's no longer cured by that drug. Right. Yeah.
Years ago, they used to recommend, if you were going into an area where malaria is prevalent, to take medication pills
Yes.
beforehand. Do they still do that? And or do they recommend taking the vaccine? And is the vaccine available for the general public?
The vaccine is available. They recommend it particularly for children, right? I don't know how available it is. In terms of the prophylactics, I certainly took it when I traveled into the Amazon jungle a number of years ago. They recommend it if you're going into an area where you're going to be spending a fair amount of time, especially if you're going out in the evenings into areas where you're going to find these mosquitoes. A firefly mangrove swamp tour kind of thing. The mosquitoes are more active at night or in the evening like that. if you were going to get malaria and you're doing that in a malaria-prone region, that might be a problem. But they definitely do still prescribe it. They'll do it specifically for, well, like I said, I had to take it for 3 weeks after I came home and like a week beforehand. So you get like the I was taking Lariam, which is 1 pill a week. Had some really weird dreams on it. But you want to take all of the pills at the right time so that you have at least more resistance to it. They're not perfect, but they're pretty good. Yeah.
I missed the beginning, but I assume you're a biochemist. What you were saying on the metabolic engineering.
Mm-hmm.
So I have 2 questions on that. One, it must be very interesting for the future to use artificial intelligence now to modify the way of the enzymes and then adapt them and take other ones. So the future will be more bright and much more quick for both the medication and the vaccine development. If you can give a bit more of your own idea on the future of these kind of developments. My other question...
Okay, that's a really... Can I answer that question? Because that's a big question. Okay. Vaccine development is actually, depending on the type of vaccine you're making, if you're doing an RNA-based vaccine, they're relatively easy to make. The long-term problem is the testing. And no AI in the world is going to get around that testing period. And that is actually where the slow process of developing drugs is big slowdown. And so I don't think AI is going to be a huge influence there. Now, where AI will be useful, I think, is in, like you were saying about these metabolic charts. Okay, these are the it can help you really quickly figure out these are the enzymes that we can change, these are the ones we have to keep. You don't want to change this one because the yeast dies, and it'll speed up that process some.
Very much.
Very much. So second question.
Yeah, I would like you to talk on the Plasmodium vivax, how it is going up north because of climate change. To Europe and other northern areas on the globe. And people should be much more aware, and that's one of the effects of global climate change.
There are a lot of diseases that are expanding their ranges just because their host species are expanding as well. I'm not, the Plasmodium vivax is one of those other Plasmodium species. It's one of the minor ones, so it's not one of the current major ones, but it's still carried by this strain of mosquitoes. It likes nice, warm, moist climates with hot temperatures and warm waters, stagnant waters. You're expanding these areas where it can grow, the disease starts becoming more common. And she's right, if the disease if these mosquitoes and the Plasmodium species become more established in places like Southern Europe, I'm going to say Greece, but I'm making that part up.
We had it earlier in Europe.
Yeah.
It got I did malaria programs in Africa for the UNDP and etc. So I read a lot on it.
You know more than I do about those parts then. I haven't read about it, but it makes perfect sense. And, organisms are changing their distribution. There is some evidence that we are going through another mass extinction because of the number of organisms that are dying out because of climate change. But again, in terms of the malaria, if you expand the ranges, you expand the range of the disease, you'll have more people get affected by it, and quite possibly more people dying from it, or resistance developing more quickly. Oh, I think John has a question. But I'd love to talk to you afterwards, because
You mentioned that in areas like Africa, it's endemic. You're not going to use these kinds of prophylactic drugs over a long term. Is our sojourn into these areas long term?
No.
Or are we okay with...
No.
prophylaxis?
We are okay with prophylaxis. We're also, unless you're going out a lot in the evening, spending time in the more stagnant areas, you are further away from the source of the mosquitoes. So sleeping on the ship with the air conditioning and that, the mosquitoes don't do quite as well, right? So again, if people like going out and exploring these places. People go off on a week-long tour going into the interior. Things happen. And if you're traveling through a malaria-prone area, hopefully you're taking at least some precautions. And, you don't want to get malaria if you can avoid it, right? But if you're going into endemic places and you're not taking all of the precautions you can, it can happen. And I'm trying not to minimize the effect of it because, you don't want to get malaria, but, if you're traveling in those areas, yeah, you've got to be aware of it.
Wow. Thanks very much, Frank. That's very helpful for all of us. You can just drink tonic water with lime and enjoy it. You don't have to put gin in it, but whatever.
Extra gin, it makes it go down easier.
And you don't care if you hurt. Okay. So let's thank Frank for his wonderful presentation. Thank you, sir.